Our next speaker is it's a local speaker from Karolinska Institute, and she's postdoc, and she's interested in how the similar SABs and Jacob impact, how the disease spread over the networks, and Annegret.
Thanks for having me. And as was already mentioned, we'll be very much aligned with Jake's talk, just in a different context. I had a little bit of an advantage over Jake because I've seen him and admired his talks at several conferences before. This is definitely inspired by it, but in another disease condition. And so I hope it's not too much alike. I try to work a little bit around it, but even if you find it too similar, please enjoy the animations. So what I am wanting to talk about, the context is not Alzheimer's, but Lewy body continuum, which usually starts very early in the clinic out with REM sleep behavior disorder on its own, which is characterized by a missing of atonia in REM sleep. So people are acting out their dreams in the REM sleep. And this is usually the first sign, and then it's very often that they progress to Parkinson's disease, 45%, 5% of REM sleep behavior disorder patients staying in only REM sleep behavior disorder, 45% progress to dementia with Lewy bodies, and 5% unfortunately die in the next 5 years. So the next step would be Parkinson's disease, where we add additionally motor problems to the disease. And this further progresses then to further cognitive decline, and not only this executive problems especially, but also visual hallucinations, and those cognitive is not a steady decline, but it fluctuates a little bit. So if it starts with Parkinson's disease and then develops motor cognitive symptoms, it's usually Parkinson's disease with dementia. If it starts with dementia and only adds motor problems after a year of having cognitive problems that are clinically detectable, it's dementia with Lewy bodies. So having said that, we are going back to the topic of the talk, which is spread of epidemics. And I think in the last autumn and the spring, there were a lot of 4 or 5 years anniversaries of a big thing happening. And I think you will remember quite a few of those concepts, because there are 2 factors that usually, and Jake already introduced a lot of them, are determining whether you are getting infected. First one is proximity, so you're close to someone infected, making it more likely that you're also getting the disease. Second factor is vulnerability of you yourself. This might be you wearing a mask or not, or you having a good working immune system. So you can approach someone or being very proximate to someone, not getting infected. If this vulnerability increases, you might also get infected. So those are the 2 factors that I want to talk about in this talk, also slightly progressing until we actually have this spread of epidemics. And we are not, especially here, talking about spread from person to person. We're not aware that this is happening in dementia with Lewy bodies in this continuum, but the spread in the brain again. So Lewy body pathology, eponymous are the Lewy bodies, and those are aggregates of misfolded alpha-synuclein. As in the case for tau and amyloid, alpha-synuclein in the healthy brain has a function very abundant in the brain and in other organs, especially in the brain in presynaptic terminals, where it's involved in vesicle trafficking and signal transmission. When it's under certain conditions, it can misfold, and then it gets sticky and aggregates together. So we have those huge aggregates of alpha-synuclein that are then also the main ingredient of the so-called Lewy bodies. We currently use this not quite in the clinic, but I expect in the next 2 years it's probably also making the next step in the clinics. This prion-like behavior of infecting neighbors with those alpha-synuclein misfolded protein in assay, where we use just CSF, so cerebral spinal fluid of a patient. They are also tested in blood samples that we add to a mixture of alpha-synuclein monomers, buffer, and fluorescent dye. Oops. And in patients, nothing much happens in the fluorescent. So we usually say negative for alpha-synuclein pathology. If we have this body fluid from a patient, however, we have usually this feed of misfolded alpha-synuclein, and this induces this prion-like behavior of inducing the misfolding in other neighboring particles and creating those aggregates that then has, let me quickly get point so that we also have an increase in the fluorescent signal over the threshold so that we define the patient as positive. Currently not in the clinic, but probably soon making this transition. So then additionally also mentioned by Jake is the transneuronal spread. So it's not staying where it is, but usually we have, in the case of Lewy body pathology, 3 epicenters, which is brainstem, amygdala, and olfactory bulb. And from there, during the disease progression, it spreads further and further, impacting more and more brain regions that then have the disease in postmortem samples, which this is where we were behind Alzheimer's disease. We currently only have a direct measurement of the alpha-synuclein pathology in histopathological samples post-mortem, but we don't have a tracer yet in PET where we directly measure the alpha-synuclein pathology in vivo in the patient. What we use an indirect measure is then neurodegeneration. Here I mainly focus on structural neurodegeneration, so the loss of brain volume or thinning of cortical structures in specific brain regions. And this is an example of what our big cohort from a measure of the huge DLB consortium, the European DLB consortium, and patients from Mayo Clinic. So there's an average volume reduction in those patients. And as you might already appreciate, so the brighter the red is, the more neurodegeneration we have. And we don't necessarily find higher neurodegeneration in brainstem, amygdala, and olfactory bulb, but it's a little bit more diffuse. So we can ask the question, okay, what is happening there when we have not this random spread, but a relatively ordered one along the system, but the associated neurodegeneration that we think is correct is correlated to the pathology accumulation, which is neurotoxic, as has been shown in animal studies. What is the difference between those? And for the first step, we try to just characterize what is happening in the neurodegeneration by using a gray matter covariance network approach, where we started out with brain region and their volumes and correlated them pairwise so that we created an artificial network. And based on this, we then applied graph theory that we can apply to those, where again, the nodes, as you've heard yesterday and now also today, are the brain regions, and the connections between those would be, in this case, specifically our correlations between the volumes of brain regions. Specifically, I want to point out the modularity, because that's where we'll show an example for which has basically aspects of integration and segregation, insofar as nodes within a mode are more connected to each other than and more segregated to other nodes of other modules. So this is now a specific example. We specifically also divided our population in men and women, because we also had a question of, we know that alpha-synuclein pathology seems to be more prominent in men than in women. So we also wanted to see, is there a difference in the resulting neurodegeneration? So we also correlated things for 58 brain regions in women and men without the disease and with the disease. And with the higher and warmer colors, you see more correlation between those regions, which we interpret as degeneration in conjunction. So there's probably, at least according to our hypothesis, something going on that is similar between those brain regions so that they're affected together at the same time. This is cross-sectional though. So here we have our modules in different colors. And what I would like you to appreciate is that in healthy men, we have a very distinct separation between cortical regions and non-cortical regions, including brainstem, subcortical regions, cerebellar regions. And interestingly, since the alpha-synuclein pathology starts from brainstem and those subcortical regions, we assume there is already some neurodegeneration that's common, a process that's common to these regions, so that they are more vulnerable in men. While it's then a little bit aligning more in women and men with DLB. So we have apparently some vulnerability in a specific sex also, not only to a specific disease, but also based on demographics of the patient. So now we are more, apart from those artificial correlations and connections that we did with the gray matter networks, we can of course also look at specific white matter connections, which are actually the roots, at least according to our hypothesis, that the pathology would use to spread through the brain. There's a nice animal study where they injected alpha-synuclein seeds, so the misfolded one, in the substantia nigra, and then waited a little bit for a few months, and then dissected those brains and looked where those pathology of the alpha-synuclein would be found after this time. As you can see here, it's quite a wide range spread, not necessarily only proximate as in very next to each other, but also proximate as in connected to each other, as you can see here in those regions where it's spread from here along the white matter tracts. We tried to replicate this similarly in DLB, where we now used and focused on the white matter tracts using diffusion tractography, building this white matter network, again, warmer colors, more connected, and thus really structural connections, not only correlations. And we hypothesized that regions that would be connected to either closely to one of the origins, here we only tested brainstem and amygdala because we didn't have a nice brain region in this atlas for the olfactory bulb, and brainstem. And generally also we assumed integrated in the system, because we assumed if you are more integrated, you have more ways that you can be reached by the pathology. So our results look like this, where we did not find any confirmation for our hypothesis for brainstem or general integration in the system, but for the amygdala, and so far as the shortest path links, so our connections are the shortest from a specific brain region to the amygdala, was predictive of having more atrophy, so a lower volumetric W score that was corrected for sex, age, and total intracranial volume. And that was also similar in the module, where the module including the amygdala, so this green module here, also had the highest atrophy in total compared to the other brain region in other modules. Now we switch gears a little bit, where we first looked at proximity, now we look at local vulnerability to the pathology, where there's two kind of opposing factors. We have clearance of the pathology and the normal alpha-synuclein that should predict lower atrophy because we are getting rid of the toxic pathology. And the opposing thing is favoring accumulation, so production, and generally keeping of the pathology should predict more neurodegeneration. So far I only talked about alpha-synuclein, but because this is supposed to be the main pathology in Lewy body disease. However, as we found is even in DLB, so in dementia with Lewy bodies, when we test them in PET or CSF, 60% have at least either tau or amyloid, so coinciding pathology in vivo, but also this was confirmed in a pathology study in Amsterdam, where we have, of course, a high Bragg stage of the alpha-synuclein pathology, but also high tau phases and Serret scores, as well as Bragg neurofibrillary terminal scores in the progress of the continuum. And that's why even the newest criteria for the diagnosis of clinically of the disease also include, if we find amyloid or tau, it's not like, does not necessarily mean it needs to be pure Alzheimer, but it's just a different mixture of pathologies. And they probably don't only coexist, as you can see here, they're co-localized. In green, we have alpha-synuclein pathology, and in red, tau. Here along a neuroid, and then granules of tau positivity here. But there's also a hypothesis that they co-aggregate in possibly two forms. Co-aggregation, so they are using monomers from both pathologies and creating those aggregates, or one might use the seed of the other and then create, for example, a tau seed creates then an alpha-synuclein pathology aggregate. This was also confirmed in a rodent study, where I mainly want to focus here on this right figure, where you can appreciate that if we have just alpha-synuclein pathology, it might aggregate and we have an increase in the fluorescent signal. But if we also have tau, this is a little bit steeper and quicker, this aggregation. So probably confirming one of those processes going on and influencing each other, being worse generally for the patient. Also clinically, they are having a quicker decline in their impairment. And this influence of different pathologies also led us to this study, where we used, again, Allen Human Brain Atlas, and not only focused on one pathology and how a gene expression might determine whether it's expressed and pathology is existent or not yet pathology is specific. Alpha-synuclein, for example, is existent, or tau or amyloid is existent, but also each other, influencing each other. We specifically used our previous atlas of 58 brain regions and defined for each of those brain regions 12 genes that are related to amyloid, tau, and alpha-synuclein aggregation and clearance. And of course, we then also used from our DLB patients the W scores of the volumes, saying negative scores against atrophy, positive scores, higher volume compared to healthy controls, the same figure that you saw previously, and then correlated those two here, just an average gene expression of healthy controls needs to be remarked, and the average atrophy pattern of DLB patients. What we found is generally, what you can appreciate here in the expression of those 12 genes, that they are relatively diversely expressed across brain regions. Of course, there is a clustering of subcortical brainstem regions, cerebellar regions usually, but of course it's an interaction between those genes. And then of course we have the atrophy pattern. When we then look at all those correlations on their own and correct for multiple comparisons, we only found a positive correlation of P-sen that is involved in the production of amyloid and those local volumes of the 58 brain regions. But then when we combine them, all those genes to predict atrophy in a given brain region in a random forest model, we also saw that it's not only one brain region, one gene expression that determines it, but it's probably an accumulation of different genes that drive those processes. Now, combining this, again, in this epidemic spread modeling, as Jake already pointed out, is we, of course, not only have a network vulnerability and a local vulnerability, but we have both in conjunction. And then we, of course, have those 5 steps where we synthesize normal alpha-synuclein over different steps of time and accumulating now in one brain region. That was an aspect of the local vulnerability. Then this might misfold. And of course, the more alpha-synuclein pathology, if I'm giving it a certain probability of misfolding, the more alpha-synuclein pathology I have, the more it's likely that one of them is at least misfolding and then starts this process. And then, of course, we have the clearing of other that is determined by another set of genes, depending always on the gene expression. Here for the alpha-synuclein pathology, it's usually SNCA that is related to the production, GBA to the reduction of alpha-synuclein pathology. And then, of course, when we now put it into context of other brain regions, things can spread along those existing connections, especially, of course, when we really define them strictly according to white matter connections. Then, of course, they can start one spread of those pathological seeds, also start those pathological processes in different brain regions. And what we last but not least define is the more of those pathological seeds we have, or the more disconnection from other brain regions we have. So it's a functional vulnerability, and one brain region gets isolated, and that's very common in DLB, that we have a disconnection syndrome. We lose connections probably earlier than actual volume. So then connection also determines. It's a little bit of a case of burning bridges. And this has been shown not only in Alzheimer, but also that this works in alpha-synuclein pathologies, here in REM sleep behavior disorder, as well as in Parkinson's disease. So if we induce the seed in silico in a specific brain region and then model the spread, as well as all those 5 steps in conjunction, we get a quite good fit with the CN atrophy in the patients, both in REM sleep behavior disorder, even there where we don't have so much atrophy, but also in Parkinson's where the atrophy is more pronounced. And then, of course, also already touched on by Jake, it's not only gene expression, but also other factors. Here it's another study by the same group in Montreal that also looked in gene expression, but also like the cell composition that are specifically protective and clear the pathology from those regions. So the more endothelial cells or oligodendrocytes you have, the cleaner your brain region is, the less likely it is to show atrophy. And that would already be the summing up of it by saying, I hope I've showed you again that there's different aspects to predict vulnerability of a brain region to atrophy, depending on whether there's a specific pathology, but also a conjunction of pathologies, and different steps need to be taken into account in order to understand whether we can predict pathology. This, of course, can then be applied clinically to say, okay, maybe we know this is the brain region possibly, hopefully soon. We also have a tracer for alpha-synuclein. There is the alpha-synuclein pathology in this brain region. We have characterized it with genetic expression and cell composition and know how much is likely to spread to another brain region. Then we, of course, can hypothesize that a given brain region has a certain function so that we can especially boost a specific function in a patient to actually give them a little bit of reserve that we can lose the specific function. So, and with this, I just would like to show my colleagues here at Karolinska Institute, and thank you for your attention.